In 2015, a deep-sea research team lowered a camera into the darkness of the Indian Ocean. They weren't expecting much. Deep-sea surveys rarely deliver on the first dive. You get sediment, you get the occasional fish, you get a lot of nothing. But at around 1,700 meters down, something moved across the frame. The team froze. It had a shell, segmented, domed, with 10 legs and eyes mounted on stalks that looked almost laughably primitive, like something a child might draw if you asked them to design an animal. One of the researchers leaned toward the monitor and said something that stuck with everyone in that room. That shouldn't exist anymore. And he was right. What they were looking at was a creature whose body plan had not meaningfully changed in approximately 500 million years. 500 million years of mass extinctions, asteroid impacts, ice ages, continental drift, and this thing just kept going, kept living, kept being exactly what it always was. The question isn't just how it survived. The question is why evolution itself seemed to leave it alone. Before we dive in, a quick note. Some of what we cover today involves genuinely strange evolutionary biology, ancient extinctions, and creatures that exist at the very edge of what we understand about life on Earth. We're going to treat all of it seriously. Welcome to Deep Extinct, where we cover the most extraordinary animals the planet has ever produced, living and gone. If that's your thing, subscribe. Let's go. Let's start with the animal that opened this story, because most people have never heard of it, and that's a genuine injustice. It's called the Limulus polyphemus, the horseshoe crab. And before you click away thinking you know what a horseshoe crab is, I promise you don't know this part. The horseshoe crab is not actually a crab. It's not even a crustacean. Its closest living relatives are spiders and scorpions. It belongs to a subphylum called Chelicerata, a lineage so ancient it was already diversified and widespread before the first fish had evolved a backbone. The oldest horseshoe crab fossils we've found date to approximately 450 million years ago. The species alive today, the ones washing up on Delaware beaches every spring, the ones being harvested for their blood in pharmaceutical labs, are, in terms of their fundamental body plan, almost indistinguishable from those fossils. Think about what was happening on Earth 450 million years ago. There were no land animals, no trees, no insects. The continents looked nothing like they do today. The oceans were filled with creatures so alien we struggle to classify them even now. Trilobites dominated the seafloor. The first-jawed fish were just beginning to appear, and horseshoe crabs were already there. Already doing what they do, already essentially what they are today. 5 mass extinctions have swept across this planet since then. The one that killed the dinosaurs. The Permian extinction that wiped out 96% of all marine species. Events so catastrophic, they reset the entire trajectory of life on Earth. The horseshoe crab survived all of them. But here's where it gets strange. It's not just that they survived, it's that they survived without changing. Virtually every other lineage that made it through those extinctions was reshaped by them, forced into new body plans, new behaviors, new ecological roles. Evolution is relentless. It never stops pressing, except, apparently, on this. Scientists call it living fossil status, a term that's somewhat controversial in biology because it implies evolution has stopped, which isn't exactly right. What it really means is that the selective pressure to change has been so low for so long that the animal has remained in a kind of evolutionary equilibrium. It works so well in so many conditions that nothing has been able to outcompete it or force it to adapt into something different. But that explanation, satisfying as it sounds, doesn't fully hold up, and we'll get to why in a moment. First, let's talk about the blood. Horseshoe crab blood is blue. Not metaphorically, not as a quirk of lighting. Literally, genuinely, vibrantly blue, the color of a clear sky in the middle of the afternoon. The blue comes from a copper-based molecule called hemocyanin, which carries oxygen through the blood the way iron-based hemoglobin does in our blood. But the color is almost beside the point, compared to what else horseshoe crab blood can do. In the 1960s, a researcher named Frederick Bang noticed something extraordinary. When he exposed horseshoe crab blood to bacterial toxins, specifically a type called endotoxins, produced by certain gram-negative bacteria, The blood clotted almost instantly around the contamination. It formed a physical barrier, trapping the toxin before it could spread. This wasn't just interesting, this was potentially revolutionary. Because detecting endotoxins in medical equipment, injectable drugs, and vaccines is one of the most critical challenges in pharmaceutical safety. These toxins are invisible. They don't smell. They don't change the appearance of a solution. But if they get into the human bloodstream, they can trigger septic shock. They can kill. Before horseshoe crab blood, detecting them required injecting rabbits with a test substance and watching for a fever response over 24 to 48 hours. Slow, expensive, imprecise. Horseshoe crab blood detects endotoxins in 45 minutes, with extraordinary sensitivity, down to concentrations of 1 part per trillion. Today, virtually every injectable drug, every vaccine, every surgical implant that enters the human body is tested using a compound derived from horseshoe crab blood called LAL, Limulus amebocyte lysate. Every IV bag, every hip replacement, every dose of every vaccine you've ever received. The horseshoe crab has, arguably, saved more human lives than almost any other animal on Earth. And this is where the story takes a much darker turn, because we harvest them. Every year, hundreds of thousands of horseshoe crabs are collected from beaches along the American Atlantic coast during their annual spawning migration. They're transported to pharmaceutical facilities, where up to 30% of their blood is drained. Then they're returned to the ocean. The mortality rate from this process is disputed. The industry says it's low, around 15%. Independent researchers put it higher. Females, which are larger and therefore more valuable, are harvested disproportionately, which hits the breeding population hardest. And horseshoe crabs reproduce slowly. They don't reach sexual maturity until they're around 10 years old. Females lay eggs only during specific full moon tides in May and June. Their recovery rate, if populations decline, is measured in decades. A creature that survived 5 mass extinctions is now genuinely threatened by the medical industry that depends on it to function. To this day, a synthetic alternative called RFC exists and has been validated by regulatory bodies in Europe and parts of Asia. The FDA has been slower to adopt it. The debate about switching continues while the harvest goes on. Before we move on from horseshoe crabs entirely, there's one more thing about them that deserves its own moment: their eyes. Horseshoe crabs have 9 eyes. 9. 2 large compound eyes on either side of their shell. 5 simple photoreceptors along the top, and 2 more near the mouth that may detect UV light. The full visual system stretches almost completely around their body, giving them nearly 360-degree coverage. But the compound eyes are the ones that changed neuroscience. In the 1930s, a researcher named Haldon Keffer Hartline began studying how the horseshoe crab's eyes process visual information. What he found was so fundamental to understanding how vision works in all animals, including humans, that it won him the Nobel Prize in Physiology or Medicine in 1967. The horseshoe crab eye has unusually large, easy-to-access nerve fibers connected to its photoreceptors. This made it the perfect model for studying lateral inhibition, the process by which the eye sharpens contrast at edges, allowing the brain to perceive outlines more clearly. This mechanism underlies how your own eyes work right now, reading these words, distinguishing the letters from the background. A creature 500 million years old taught us how our own vision functions. Think about that for a second. And it raises a question that evolutionary biologists still find genuinely difficult to answer with full satisfaction. If an animal is this useful, this perfectly engineered for what it does that explain why evolution left it alone? Or does the explanation run deeper? We'll get to that. But first, the horseshoe crab isn't the only one. Because here's the thing, the horseshoe crab is the most dramatic example, but it's not alone. The deep ocean, and some very specific environments on land, have been quietly harboring creatures that look like evolution forgot to send them the memo. The Nautilus. A cephalopod, related to squids and octopuses, living in a coiled external shell that has remained essentially unchanged for approximately 500 million years. while its relatives evolved into the most intelligent invertebrates on Earth, developing complex camouflage, jet propulsion, and remarkable problem-solving ability. The Nautilus stayed in its shell, drifting, feeding on carrion and small crustaceans, living slowly, with a metabolism so low it can go months without eating. It has survived because the deep water it inhabits is stable, cold, dark, and largely unchanging across geological timescales. The selective pressure that drives adaptation requires instability, environmental shifts that punish the old way of doing things and reward the new. In the Nautilus's world, the old way never stopped working. Then there's the coelacanth, a fish thought to have gone extinct 66 million years ago, wiped out in the same event that killed the non-avian dinosaurs. For decades, the coelacanth existed only in the fossil record. Paleontologists used it as a reference point for Devonian life. It was a closed book. Until December 1938, when a South African museum curator named Marjorie Courtenay-Latimer noticed something unusual in a pile of fish brought in by a local trawler. A large, steel-blue fish with strange, fleshy, limb-like fins. She didn't know what it was. She sent a sketch to a fish expert named J. L.B. Smith. He knew immediately. His response, documented in letters that still exist, was something close to stunned disbelief. The coelacanth was alive, not fossilized, not extinct. Alive, in the deep waters off the coast of South Africa, looking almost exactly as it had 66 million years ago. The discovery shook the scientific world in a way that's hard to overstate. It meant that extinct was not always a permanent state. It meant that the deep ocean could hide things, large things, significant things, for geological timescales without us having any idea. And this next part might be the most unsettling of all. The coelacanth's fins. Those strange, fleshy, lobed appendages that make it look like it's using its fins as proto-legs. They are structurally similar, not identical, but similar, to the fins of the ancestors of all land vertebrates. Of every amphibian, reptile, mammal, and bird that has ever walked, crawled, or flown on this planet, including us, looking at a coelacanth is looking at a distant, imperfect mirror of what our own deep ancestors may have resembled 375 million years ago, before they crawled out of the water and began the long journey toward everything that came after. We'll get to what this pattern means, why these creatures cluster in specific environments, and what that tells us about life itself. But first, let's go back to the deep ocean. There's a pattern running through all of these creatures: the horseshoe crab, the nautilus, the coelacanth. And it points to something profound about how evolution actually works. They all live in, or retreat to, environments that are extraordinarily stable across geological time. The deep ocean, below the thermocline, roughly 200 meters down, exists in a kind of suspended permanence. Temperature barely fluctuates. Pressure is crushing and consistent. Light doesn't reach it. The chemical composition of the water changes on timescales of millions of years, not thousands. For a creature adapted to this environment, there is almost nothing forcing change. Evolution is not a relentless drive toward complexity or perfection. It is a response to pressure. Remove the pressure, and change slows or stops. An animal that is perfectly adapted to a stable environment, with no predators it can't escape and no competitors it can't outlast, has almost nothing selecting against its current form. This is why researchers now prefer the term stabilized lineage over living fossil. It's more accurate. These animals are still evolving. At the molecular level, in their immune systems, in subtle biochemical ways we're only beginning to detect with modern genomic tools. But their bodies, their visible form, the architecture that defines what they are, that has been locked in place by millions of years of successful survival. But here's where it gets strange, because the stability explanation doesn't account for everything. Horseshoe crabs, for instance, don't only live in the deep ocean. They come ashore to breed. They live in estuaries, in bays, in relatively shallow coastal water. Environments that have changed dramatically over 450 million years. Coastlines that have shifted, sea levels that have risen and fallen by hundreds of meters. Ocean temperatures that have swung through ice ages and warming periods that would have reshaped their habitat completely. And yet, they remained. Some evolutionary biologists argue that the horseshoe crab's survival isn't just about environmental stability. It's about a body plan so fundamentally robust, so flexible in the range of conditions it can tolerate, that it simply never encountered a challenge it couldn't handle without redesigning itself. Others point to something harder to quantify: luck. The role of chance, in which lineages survive mass extinctions, is significant. The asteroid that killed the dinosaurs didn't kill them because they were less fit than mammals in any absolute sense. They were wiped out by a specific, catastrophic event that mammals happened to survive by virtue of their size and behavior. In a slightly different scenario, the roles might have been reversed. The horseshoe crab survived 5 of those scenarios. At some point, that stops being luck and starts being something that deserves a better name. Here's what keeps deep-sea biologists genuinely awake at night. We have explored less than 20% of the ocean floor. In terms of direct observation and detailed survey, the kind where you can actually see what's living there, it's more like 5%. The deep ocean is the largest habitat on Earth. It covers more area than all of the planet's land surface combined. And we have barely looked at it. The coelacanth was hiding in plain sight, in a relatively well-traveled stretch of water off South Africa's coast, for 66 million years. We only found it because a museum curator happened to recognize an unusual fish in a pile of trawler catch. How many other lineages, equally ancient, equally unchanged, are living in the parts of the ocean we haven't looked at yet? This isn't a rhetorical question. Researchers take it seriously. Every few years, a deep-sea survey pulls up something that doesn't fit the existing record. In 2021, a survey off the coast of Western Australia found more than 30 species previously unknown to science in a single expedition. A coral reef system the size of the Empire State Building was discovered off the Queensland coast in 2020, taller than any known reef structure on Earth, completely undetected until a routine mapping survey passed over it. The ocean is not a known quantity, not even close, and the creatures living at its deepest, most stable points have had 500 million years, 500 million years, to perfect the art of persisting without being noticed. To this day, researchers still can't fully explain why the patterns of extinction that have reshaped every other major animal group on Earth have bypassed these lineages with such apparent indifference. The honest answer from the scientific community is that we don't have a complete model. We have contributing factors. We have hypotheses. We have a growing genomic picture that is revealing layers of molecular adaptation invisible to the naked eye. But the full story of how a creature survives 500 million years without becoming something different, that story is still being written. Here's the thought I want to leave you with. When we talk about evolution, we usually talk about it as a story of change, of diversification, of lineages branching, adapting, becoming something new. And that story is true. But the horseshoe crab, and the nautilus, and the coelacanth, and the handful of other lineages like them tells a different story. A story about persistence, about the extraordinary power of a design that works so well in an environment stable enough to let it keep working, that 500 million years can pass, and the answer to what is this thing remains essentially the same. These animals are not relics. They are not failures to evolve. They are, in their own way, the most successful organisms that have ever lived, measured not by complexity, or intelligence, or ecological dominance, but by pure, stubborn, extraordinary duration. Everything we think we know about the history of life on Earth has been assembled from fragments: fossils, genomic data, living specimens, deep-sea surveys that have covered a fraction of what exists. The living fossils remind us that the fragments are incomplete, that the ocean, the same ocean that has existed in some form since this planet had water, is still keeping secrets. And perhaps the most unsettling question of all, what else is down there, unchanged, undetected, simply waiting for us to finally look in the right place? Subscribe to Deep Extinct for more. Drop a comment below. Which of these creatures gets to you the most? The horseshoe crab that's saving lives? The coelacanth that came back from the dead? Or the idea that the deep ocean might be full of things we haven't found yet? You don't want to miss that one.